So, welcome back, and we will continue our discussion about the B cell activation and differentiation inside the lymph node and in the germinal centers of the lymph node. So, what are the events in the germinal center of the lymph node happening? So, as we discussed in our last class, if you look into the picture from our last class, we have discussed about the activation of the B cell, primarily the thymus dependent activation. We have discussed much more in details. So, when there is a thymus dependent activation, and this process is primarily meant for example, the protein antigens, the proteins, the protein antigens that are processed antigens. So, these protein antigens, they actually elicit this kind of a response. That is a thymus dependent response, and the thymus dependent activation, as we have seen, that it leads to proliferation of the B cell. Now, the B cell, we call it the activated B cell. So, here after this B cell here, this B cell here is now it is an activated B cell, because it has got signals from the T follicular helper cell, the follicular helper T cell. It has received the signals for its activation, and it is now kind of ready to develop into a specific B cell producing a specific antibody type against the antigen that it has captured. So, it has captured the antigen, presented it by the MHC class II to a T helper cell, and the T helper cell helps it to activate, and now this B cell starts to proliferate. Now, it starts to increase in population. So, now it proliferates where? In the germinal center. Now, this what happens in the germinal center? Let us look quickly in the next part. So, now this is the lymph node, this is the lymph node, let us say. So, the lymph node, I told you previously as well. So, the lymph node has some specific regions, the medulla, the paracortex and the cortex. So, there are at least three specific regions in the lymph node: the medulla, the paracortex and the cortex, and within the cortex you have this kind of follicular structures. These are the germinal centers. Now, these germinal centers contains this B cells, this proliferating B cells. So, once the B cell is activated, so what we got from our last lecture, these activated B cells, they will now enter into this germinal center. They will now enter into the germinal center, and they starts to proliferate here in the germinal center. So, they now increase in number, undergoes mitosis, and they will start to proliferate, increase in numbers. And at this stage, I told you they will start expressing certain chemokine receptors, and these are the CXCR4 receptors, and these receptors can recognize a specific chemokine, which is the CXCL12, which is secreted from the stromal cells, and this helps, this interaction of this chemokine to the chemokine receptor helps to retain the retains this interaction here is primarily required for retention of these centroblasts, retains the centroblasts in the dark zone. So, this is the dark zone. This part of the germinal center, we call it the dark zone. And this part where the final differentiation occurs, we call it the light zone. Now, what happens in this dark zone, these cells which has started proliferating, these are called the centroblasts. Now, these centroblasts, they will undergo a specific process known as the somatic hypermutations. So, they will undergo somatic hypermutations and they will produce different centroblasts with different affinities. Affinities for what? So, the B cell, they will develop different B cell receptors or the antibodies on the surface which has different affinities for the antigen. So, these centroblasts have this kind of, they express these antibodies on the surface which will now have different affinities for the antigen. So, they have been activated by an antigen that we have seen in our previous lecture, in the previous case. So, now they are activated, these B cells are activated, and these centroblasts, what we call the centroblasts, now will undergo what is called somatic hypermutation. It is called somatic hypermutation. Somatic hypermutation, that is the somatic hypermutation, and during this process of somatic hypermutation they start to develop mutations. So, there are mutations in the variable region. In this somatic hypermutation process, so the somatic hypermutation process, they will start to develop mutations in the variable region. Mutations occurs in the variable region. So, there are point mutations in the variable region, and these point mutations are primarily carried out by an enzyme called the AID. So, AID is basically activation induced cytidine deaminase. It is the activation induced cytidine deaminase. So, this activation induced cytidine deaminase that leads to deamination, and then there is kind of DNA repair processes like base excision repair or mismatch repair. There are different processes of DNA repair processes that are being activated after this AID. So, we have the AID. Now, these centroblasts here, they will start expression of this AID. They will express this AID enzyme. So, this activation induced cytidine deaminase is being expressed in the centroblasts. Now, these centroblasts, they will start expressing AID and will acquire mutations in the variable region of the heavy and the light chains. So, now they will have mutations in the variable regions, and they will each of these centroblasts will now express B cell receptors on the surface which have different affinities for different for the same antigen. So, they have different affinities for the same antigen. So, some of them will now carry high mutation, high affinity or low affinity. So, this hypermutation is primarily to increase the affinity of the antibodies towards the antigen. So, now these centroblasts which are now prepared to enter the light zone. So, these centroblasts are now kind of prepared to enter the light zone. They will now start to express on their surface and the class of chemokine receptors, which is the CXCR5. Now, what is the role of the CXCR5? Let us see. So now what happens? Now these centroblasts in the dark zone, they have proliferated, undergone somatic hypermutation, point mutations being introduced in their in the variable regions of their heavy and light chains. So now they have differentiated into, now they are different, they are not same. So each of the centroblasts, so maybe some centroblast, for example this one, let us say here, this has very high affinity for the antigen. The receptor has a very high affinity for the antigen, and let us say this one does not have a high affinity, so its affinity has reduced towards the antigen. So they have either a good mutation or a bad mutation. So if it is a bad mutation, that means a low affinity mutation, it's the receptor's affinity towards the antigen is reduced. If it has a good mutation, then its affinity towards the antigen has increased or improved affinity. So there can be two situations, it has bad or low affinity, or it has good or improved affinity. Now these cells, for getting selected for the affinity, now these cells has to be selected, because they have to be selected for their affinity. So which one has higher affinity, which one has more affinity, so that has to be selected, only those cells will be selected, and those cells which out of this somatic hypermutation which has developed lower affinity, whose affinities has reduced, will now be rejected. Now these cells has to come into the light zone. So what is there in the light zone? In the light zone we find specialized cells like the follicular dendritic cells. They have this kind of structure, and these dendritic cells, they are the primary producers of a class of cytokines, that is the chemokines, basically chemokines, that is the CXCL13. Now this CXCL13 is a ligand for this CXCR5 receptor. So this CXCL5 binds to this CXCR5 binds to the CXCL13, and they are attracted towards the, they are kind of attracted towards the light zone. So now they start moving into the light zone. In these centroblasts, they start moving to the light zone now. Now once they come to the light zone, they are the centrocytes, we call them the centrocytes, and these centrocytes, they, as I told, they can have a high affinity mutation or they can have a low affinity mutation, and as per that mutation they will have different fates. So now they can have two different fates. Let us say this one has a high affinity mutation and this one has a low affinity mutation. So what will happen? Now this one which has a lower affinity mutation, that means now it has less affinity towards the antigen, will undergo apoptosis. And there are specialized macrophages in this region of the light zone, in this in the germinal center. There are a specialized group of macrophages who will engulf them. These apoptotic cells will be engulfed by a specialized class of macrophages, and they will die. So they will not exist, they will be excluded from the system. These low affinity mutation cells, the cells which has low affinity mutations on their B cell receptors. Those with the high affinity receptors will now be helped by these follicular dendritic cells. Now what does this follicular dendritic cells do? So these follicular dendritic cells, they kind of adheres these antigens on their surface. So they now display these antigens on their surface. These antigens are present on these follicles on the surface of these dendritic cells. This is the antigen. So now these B cells or these centrocytes, these are the centrocytes. These centrocytes which has a high affinity, Now, since it has a high affinity, it will try to capture one of these antigens by the B cell receptor. So, a B cell will now interact by this B cell receptor with this antigen, which is being adhered to the surface of the follicular dendritic cell. So, this is the follicular dendritic cell, we also call them the FDC or the follicular dendritic cell. This follicular dendritic cell is present inside the germinal center, inside the light zone of the germinal center, and they carry these antigens. So, they carry the antigens on the surface, and these B cells, the activated B cells, which has already a very high affinity mutation. That means they now have developed higher affinity for the antigens. They will interact with the antigens that are captured on the surface of the follicular dendritic cells. So, now they will immediately get hold of this antigen by these B cell receptors, and again there is another interaction. What is that interaction? So, now they will interact with the T helper cells. And which class of T helper cells? They will interact with the T follicular helper cells again. So, there are follicular helper cells that are present. So, this is a B cell. Now, again these B cells which has interacted with, which has the high affinity and has interacted with an antigen with a very high affinity, they will now again internalize. So, they will now again internalize the antigen, process it, and again present it on the surface by MHC class 2. So, then again there is a MHC class 2 interaction. So, they will now present the antigen on the surface by MHC class 2, and again there are follicular helper TFH cells that are present here, the T follicular helper cells, the TFH or the T follicular helper cells which are present in the light zone of the germinal center. They will interact by the T cell receptors. So, with the TCR, so there will be an interaction, and again there will be interaction of the CD40 ligand to the CD40. So, again a CD40 to CD40L interaction. The CD40 ligand which is expressed on the TFH cell will again interact with the CD40, and this interaction here is very essential. So, this interaction here is very essential for the next step, that is the class switching. So, let us look at this part again quickly. So, there is somatic hypermutation leading to increase or decrease in the affinity of the B cell receptors expressed on the surface towards the antigen. Remember, then these B cells, the centroblasts, they will start expressing CXCR5. CXCR5 has high affinity towards CXCL7. This is a chemokine. A chemokine called the CXCL7 is produced from the follicular dendritic cells, the FDCs, and that will pull that will bring due to the attraction, these centroblasts will now move to the light zone, and now they are called the centrocytes. Now, these centrocytes either can have a high affinity or a low affinity. Now, then there it needs a selection. So, then there is an affinity maturation or an affinity selection process, and this process is called affinity maturation or affinity selection. So, now depending on its affinity towards the antigen, it will be selected. So, either if it has a low affinity, bad affinity or a low affinity, it will undergo apoptosis. If it has a high affinity, it will be exposed to the antigens that are presented by or that are captured by the follicular dendritic cells that are present here. Now, this follicular dendritic cells which has captured antigen on the surface will present this antigen to this centrocyte or the B cell or the high affinity receptor producing B cell, and these high affinity receptors can now bind with very high affinity to this antigen. And this binding ensures that the B cell is now, this centrocyte is now selected for a high affinity. So, this process is known as affinity maturation or affinity selection. So, now the high affinity B cells or the B cells having very high affinity towards the antigen will be selected out of the whole population. It is a very small population, it is not a very big population. So, many of the cells they die by apoptosis, and once they die, these apoptotic cells are also phagocytosed by a class of macrophages that are present here. So, they will be killed, basically they will be rejected. That population of the B cell is rejected. So, in this whole process of this immune system, there are very careful checkpoints everywhere. So, it is not like the processes are very random. So, there are very well designed checkpoints where it is being kind of checked, or there are quality control checks. So, there is a mutation, there will be a quality control check. The B cell receptor with the high affinity will pass through, with the low affinity will not pass through, they will be dying, they will be rejected, and that population will not continue to exist. So, the high affinity population will be selected and by a process called affinity maturation with the help of the follicular dendritic cells and of course, the T follicular helper cells, the TFH cells which are also present in this region. Now, the TFH cells, as we remember from the last lecture, the TFH cells, one of the major components that is being secreted by these TFH cells are interleukin 21, remember the IL-21. So, now these TFH cells will start to produce IL-21 as well, interleukin IL-21, a specialized interleukin or a specialized cytokine that is being produced from these TFH cells, the follicular helper cells. So, this IL-21 will be produced, and that IL-21 is primarily required for the survival and the proliferation of the B cells, the matured B cells, the activated B cells, as well as they will start to produce a whole subset of cytokines. There will be production of a whole set of cytokines, and that will actually govern the process called the class switching. So, now these antibodies will start to class switch. So, they will preferably induce class switch to different isotypes. So, now these antibodies, so far we were still having these IgMs on the surface. The main B cell receptors are comprised of these IgM molecules. Now, they will start to switch. So, there will be isotype switching, there will be class switching, and then they will start to produce different types of antibodies. Now, they will start producing different types of antibodies like IgG, IgE. So, for example, and this is being governed by the types of different cytokines that are being produced from them. So, for example, they will produce interleukin 4, you will get a more detailed idea about this. So, different cytokines, they will preferentially they will induce the switching to different types of isotypes. For example, you will have IL-4, interleukin 4. When we will study about the cytokines more in details, we will see the role of the different cytokines, the different interleukins. So, for the time being, let us know that this interleukin 4 is mainly responsible for switching to IgE, that is immunoglobulin E, which is responsible for mainly the hypersensitivity reactions, allergic reactions. You will have IgE, you can have interferon INF gamma, which mainly switches it to IgG. You can also have IL 21, which is also responsible for IgG. So, there are many other cytokines to name. I am only naming a very few of them, I am not naming all of them. So, this is a very small class of names that I have mentioned here. We will get to know this when we will learn the cytokines more in details. For the time being, I am not discussing them in very details. So, but at least you know that once there is this kind of an interaction. So, once it is affinity selection process is done, affinity maturation process is done, that is the B cell or the centrocyte is selected for the high affinity mutation. Those high affinity cells will now interact with the T follicular helper cells, and this CD40 ligand interaction will again occur. MHC class 2 will present the antigen on the surface and will interact with the TCR, with the T follicular helper cell, leading to secretion of different cytokines. These cytokines will basically help in the class switching to different isotypes. Now, this now they will class switch, and depending on the type of cytokine that is being secreted or the type of cytokine that is available, they will switch into different antibody types. So, different isotypes. So, now this process we call it as class switching. So, this is the class switching. We are not going into the detailed mechanism of the class switching at this moment, but these cytokines, as you know, they lead to certain activation of certain transcription factors, and that can actually lead to the final effect. So, the cytokines mediate their action by mostly by the induction of or activation of certain transcription factors downstream. We will read about this more in details when we study the cytokines in details. Now, after the class switching phenomenon, now the last step is finally these cells will finally become, either they will become a plasma cell. So, either they will develop into a plasma cell, or into a memory B cell. So, it can become a memory B cell, or into a plasma cell. So, that will produce the antibodies, the soluble antibodies. So, now these antibodies will now be the mediators actually. So, this is the plasma cell, and this is the plasma cells, and finally these plasma cells will again, they will migrate to the bone marrow. So, these plasma cells will now finally again migrate to the bone marrow, and from there they will start secreting the antibodies. So, if you look into this whole picture, at least in this part, so we have at least a few distinct steps that are occurring in the whole process of activation and the differentiation. So, after the B cells are being activated, these B cells, they will enter into the germinal center, into the dark zone. In the dark zone, these centroblasts, they are centroblasts, and these centroblasts, they will start proliferating. So, this is a process of proliferation, and in this proliferation process, they will start expressing a particular enzyme known as the activation induced cytidine deaminase, which leads to point mutations that actually introduces point mutations in the variable regions and leading to a process called somatic hypermutation. This somatic hypermutation. Now, this somatic hypermutation introduces point mutations in the variable region, leading to an increase or decrease in the affinity towards the antigen. Now, this centroblasts will then cross the dark zone, they will enter into the, from the dark zone they will enter into the light zone by the action of a chemokine. Once they come to this light zone, these are the centrocytes. Now, these centrocytes will start interacting with the antigens that are captured on the surface of these follicular dendritic cells. Follicular dendritic cells capture these antigens on the surface and present it to the centrocytes. Centrocytes interact with the B cell receptor, the high affinity ones will interact with the B cell receptor, low affinity ones will be rejected and will undergo apoptosis. Now, once they are selected for the high affinity, then this once they can recognize the antigens by their high affinity receptors, they will internalize again the antigen and will present it on the surface by class 2 MHC molecules. Class 2 MHC molecules that will interact with the T cell receptors on the T follicular helper cells, which are also present in the light zone. Now, then there is CD40 CD40L interaction occurring and the TCR interaction, and that leads to secretion of certain specialized molecules or the specialized cytokines. These cytokines includes interleukin 21, as we know that typically interleukin 21 is secreted from the TFH cells, and that is required for the survival as well as the proliferation of these centrocytes, and as well they will produce certain specialized cytokines like interleukin 4, INF gamma, interleukin 21, which leads to the class switching phenomenon. So, switching the class switching to different isotypes, and now they will produce different antibodies will now be produced, they will switch to different antibodies like IgE, IgG, and then these centrocytes or these B cells will now differentiate into a memory B cell or into a plasma cell, and finally this plasma cell will again migrate back to the bone marrow. So, where we started from, so it goes back to the bone marrow and it starts secreting the antibodies which mediates all these functions. So, we got to know about in this lecture we got to know about the B cell activation and differentiation, how the B cell activation occurs, how it differentiates into different cell types, different and finally class switches into different isotypes, produces different types of antibodies, and you can go through all the details in the book, in the books that we have referred to. You will be finding more details on the different steps. I did not go into the very details of the different steps because that is not really possible in this short period of time. It is very difficult because there are a lot of molecular phenomena that are involved in the somatic hypermutations or the class switching phenomenon. So, those there are many molecular events that are occurring. So, it is very difficult to cover in this short span of time, but I try to summarize the whole events that are occurring within the germinal center leading to the activation and the differentiation of the B cells. So, that is all from the B cell maturation and development and activation part, and I hope you liked it. Thank you.